Good morning, everyone. This is Sumana, Assistant Professor of Biochemistry. Today's our topic is the concepts or terms like holoenzyme, apoenzyme, coenzyme, and cofactor. And what are the definitions? What is the role of these factors? We'll discuss. See, the holoenzyme. Holo means whole or complete. Holoenzyme, the definition for this is the functional unit of the enzyme is known as the holoenzyme, which is often made up of apoenzyme, that is the protein part, and a coenzyme, non-protein organic part. The both protein and non-protein part of the enzyme are together will be considered as the holoenzyme, which is required for the enzyme catalysis of the reactions. Okay. And the apoenzyme is nothing but the protein part of the enzyme is called the apoenzyme. The coenzyme, the non-protein organic part of the enzyme is called cofactor. The non-protein organic part of the enzyme is called coenzyme. And the non-protein of the enzyme is called the cofactor, like metal ions. See, suppose the non-protein part is organic part, that is which is consisting of the hydrocarbons in the chain, that is called coenzyme. Suppose if it is metal ions or inorganic molecules are there, it is called cofactor. Here you can see in the equation, this is the holoenzyme, which is the active enzyme. The enzyme will be active only when both protein part and non-protein part of the enzyme are together. That is apoenzyme or coenzyme are together. Then only the holoenzyme can be actively participate in the catalysis. And the term the prosthetic group is used when the non-protein moiety is tightly or covalently bind with the apoenzyme. Covalently means is the covalent bond is formed by the sharing of the electrons. So when the non-protein part of the enzyme is tightly bound with the enzyme or covalently bind with the enzyme with the apoenzyme, then it is called prosthetic group. Prosthetic group is nothing but a non-protein part which tightly bind with the enzyme. And the coenzymes can be separated by dialysis from the enzyme, while the prosthetic groups cannot be. See, the dialysis is one of the technique or procedure to separate the molecules, and the coenzymes can be easily dialyzable or can be separated with the dialysis, but not the prosthetic group, because the prosthetic group is tightly bound with the enzyme by sharing of the electron and by forming the covalent bond. And the coenzymes, see, the coenzyme definition is the non-protein organic means which is consisting of the hydrocarbons, low molecular weight and dialyzable substance. Just now I explained about the dialysis, that is coenzymes can be easily separated from the holoenzyme, that's why it is called dialyzable substance, associated with the enzyme function is known as the coenzyme. And the term activator is referred to the inorganic cofactor like calcium, magnesium or manganese necessary to enhance the enzyme activity. See, both coenzyme or cofactor are non-protein part of the enzyme, but when this non-protein part is organic, then it is called coenzyme, and when the non-protein part is like inorganic or metal ion, it is called cofactor. And when this cofactor is tightly bind with the apoenzyme is called prosthetic group. This is the difference between these terms. And coenzymes acts as are second substrates. Why? Because the coenzymes are often regarded as the second substrate or co-substrate, since they have affinity with the enzyme comparable with that of the substrate, because in the when it takes consider the enzyme also, there are active site or in substrate binding site and also the catalytic site. At the active site the substrate will get bind, and at the catalytic site coenzyme will be bind with the enzyme, and these coenzymes undergo alterations during the enzymatic reactions which are later regenerated, and this is in contrast to the substrate which convert to the product. Once the coen coenzymes are not going any change, even if they are undergoing any change after the reaction, it will comes to the original form. And the coenzymes participate in various reactions involving the transfer of the atoms or groups like hydrogen, aldehyde, keto, amino, acyl, methyl, carbon dioxide, etc. The coenzymes will be involved in the transfer of these molecules. Coenzymes play a decisive role in enzyme function. And here a few examples are there for the coenzymes which are from the B complex vitamins. See, B complex vitamins like thiamine, riboflavin, niacin, all these B complex vitamins acts as a coenzyme in the forms. In fact, the biochemical functions of the B complex vitamins are exerted through their respective coenzymes. Like thiamine. Thiamine is nothing but B1, and for the thiamine the coenzyme is thiamine pyrophosphate. Here you can see thiamine pyrophosphate. And what is the function of this enzyme is it will transfer the aldehyde or keto group. The best example for the TPP is trans ketolase. And like for riboflavin, there are two forms of coenzymes. One is FMN and FAD. FMN is nothing but flavin mononucleotide, and FAD is flavin adenine dinucleotide. The main function of the coenzymes of riboflavin are hydrogen and electron transfers, and FMN is for the L-amino acid oxidase, and FAD for D-amino acid oxidase. You can remember easily like that, FAD, D for D-amino acid oxidase. And next one is the niacin. Niacin is also available in two coenzyme forms. One is NAD and other is the NAD+. NAD is nothing but nicotinamide adenine dinucleotide, and NADP is nicotinamide adenine dinucleotide phosphate. And for the example for this niacin coenzymes are also involved in the hydrogen and electron transfer. NAD example is lactate dehydrogenase, and for the NADP is glucose 6 phosphate dehydrogenase enzyme, for which NADP will act as a coenzyme. And next one is lipoic acid, and the available coenzyme is also lipoic acid, which is also involved in hydrogen or electron transfer. The example for this is, or the enzyme for this coenzyme is pyruvate dehydrogenase complex. Next is pyridoxine. For the pyridoxine, the coenzyme is PLP, pyridoxyl phosphate, and it will transfer the amino or keto group. And the example is alanine transaminase. Next is pantothenic acid. See, in most of the reactions you will see that coenzyme A, that is nothing but which is a coenzyme form of pantothenic acid, which is involved in the transfer of acyl group. Example is thiokinase. Next is folic acid. Folic acid coenzyme form is FH4, that is tetrahydrofolate, which will involved in the transfer of one carbon, that is formyl methyl. Only a single carbon can be transferred with the enzyme coenzyme, that is FH4 tetrahydrofolate. The example is formyl transferase. Next is biotin. The coenzyme form is biocytin and it will transfer the CO2, that is carbon dioxide. Example is pyruvate carboxylase. Next is cobalamin. Cobalamin, the coenzyme form is methylcobalamin deoxyadenosine cobalamin. And the transfer groups by this coenzyme are methyl and isomerization. The best example for this is methylmalonyl CoA mutase. For this enzyme, this cobalamin acts as the coenzyme. And there are non-vitamin coenzymes also. Up to now we have discussed about the B complex vitamins which are acting as the coenzymes. There are some coenzymes which are non-vitamin in nature. Not all coenzymes are vitamin derivative. There are some other organic substances which have no relation with the vitamins but function as coenzyme. They may be considered as non-vitamin coenzymes. Examples are ATP, CDP, UDP, etc. And here in the table you can see that ATP, nothing but adenosine triphosphate. The biochemical function of this coenzyme is it will donates the phosphate, adenosine and adenosine monophosphate, that is AMP moieties. Next is the CDP, cytidine diphosphate, which is required in the phospholipid synthesis as carrier of choline and the ethylamine. You have seen that in the phospholipid synthesis there is a need for the carrier of choline and the ethanolamine. For this carrier, CDP will act as a carrier. Next is UDP. UDP is uridine diphosphate, which is a carrier of monosaccharides, that is glucose, galactose, which are required for the glycogen synthesis. Next is SAM. SAM is nothing but S-adenosyl methionine and active methionine, which will donate the methyl group in biosynthetic reactions. Next is PAPS, that is phosphoadenosine phosphosulfate, which active sulfate, which donates the sulfate for the synthesis of mucopolysaccharides. And other examples are nucleotide coenzymes. Some of the coenzymes possess nitrogenous bases, sugar and phosphate. Such coenzymes are therefore regarded as nucleotides. Examples are NAD plus, NADP plus, FMN, FAD, coenzyme A, UDPG, etc. These NAD, NADP are FMN and FAD are nothing but B complex vitamins coenzymes, but it consisting of the nucleotide. Nucleotide is nothing but which consisting of phosphate group, nitrogen base and the sugar. In these molecules, these three are there. That's why it is called nucleotide coenzymes also. And other top one is protein coenzymes. Like thioredoxin is a protein that serves as the coenzyme for the enzyme ribonucleotide reductase. So for the coenzymes, B complex vitamins, non-vitamins, nucleotide coenzymes and protein coenzymes are there. And these coenzymes do not decide the enzyme specificity. See, all the enzymes are specific in their character and the enzyme specificity is because of the active site. So, a particular coenzyme may participate in catalytic reactions along with different enzymes. For instance, NAD plus, that is nicotinamide adenine dinucleotide, can act as a coenzyme for lactate dehydrogenase and also for the alcohol dehydrogenase. This single coenzyme is acting as a coenzyme for two or more enzymes. So, the enzyme specificity is not because of the coenzymes. In both the enzymatic reactions, this NAD plus is involved in the hydrogen transfer. And the specificity of the enzyme is mostly depend on the apoenzyme and not on the coenzyme. See, the holoenzyme is consisting of two parts, that is protein part and the non-protein part. Protein part is called the apoenzyme and the non-protein part is called the coenzyme. So the enzyme specificity is only because of the protein part of the enzyme, that is the apoenzyme. And thank you to all. And here we have discussed that holoenzyme, apoenzyme, coenzyme and cofactor. Holoenzyme is nothing but the enzyme which consisting of protein part and the non-protein part. Apoenzyme is the protein part of the enzyme and coenzyme is the non-protein part of the enzyme which is organic in nature. And the cofactor is the non-protein part of the enzyme which is inorganic or metal ion. And suppose if this cofactor is tightly bind with the enzyme, it is called prosthetic group which is inseparable from the enzyme. Coenzymes can be easily separable by the dialysis. Thank you to all.